There we go. Start recording. Take it away.
Good. Okay. Well, thanks very much. Yes, you gathered from my bio that I'm a Canadian who's been sort of lost in the UK for a few years and now transitioning to be a Canadian again, just moved to Nova Scotia. And so I've really been looking forward to coming here and visiting, making some new connections with the agriculture community in Nova Scotia. So if anybody's interested and wants to talk about these ideas that I present, then please do be in touch somehow. So I'm going to talk about some ideas from natural selection and adaptation and how these can help us to understand and improve agriculture. And more specifically, I'm going to talk about my work with wheat and some new ideas of how to redesign wheat crops. Now, before I get really started, I want to tell you the story of America's Cup. I promise this is somewhat related to what I talked about.
No judgment here.
But what's really interesting about this story is the American team that was in this boat, the Liberty, had won this race, this yacht race, for 132 years in a row. So it's one of the greatest sporting events ever. But in 1983, the Australian team decided they were tired of this. They wanted to do something really different, sort of think outside the box, and they finally win this race. So one of the things that they did is they hired an artist to work on their boat, to redesign their boat. And this artist didn't really have a lot of experience with racing, but what he did is took inspiration from adaptations that he saw in nature. So one of the things that he did was to redesign the keel of the boat. He did that redesign based on the shape of a birdwing. So knowing that birdwing is well designed to flow through a fluid, he thought that would be to speed up this yacht. And so it turned out that in 1982, finally, the Australians beat the Americans. And this winged keel design has really become a revolution really in the design of yachts. So I don't really care about sailing that much myself, but I see this story as perhaps an analogy for the work that I do. And like many of you, the context of my work, and so my version of the America's Cup is this challenge of sustainable food production. So we want to have crops that do produce a lot of healthy food, but do not harm more natural ecosystems or continue to take over those natural ecosystems altogether. So ultimately, we want some kind of harmonious interaction between agriculture and more natural ecosystems. But the problem is that modern agriculture is often not at all harmonious with nature. So in the extreme, some crops can seem to be eating up huge areas of land and replacing that with very little biodiversity. And it's that kind of high intensity agriculture that also leads to the overuse of inputs like nitrogen fertilizers that end up polluting our more natural ecosystems. And so while these environmental costs have been piling up, it's also the case that some of the most important crops in the world have had yields that seem to plateauing over time. So in this example, the UK wheat yield over time really seems to be hitting a plateau within the last few decades. So we're now at a point where we really need new ideas for a sustainable agriculture. And so, like in my analogy, I guess, like the artist designing Australian boat, I think it's important to take some inspiration from natural selection and adaptations that we can find in nature. So in particular, I've spent a lot of my career studying social evolution and adaptations that organisms use for living in groups. And so I think these kind of ideas are actually crucial for understanding how to design crops. So to address this question, how do we design efficient, productive crops? I'm going to present this solution that what we need to do is exploit cooperative crop planting. And so I need to explain that this term, cooperation, in evolutionary biology, we think of a cooperative trait as one that has a beneficial effect on others, and that trait is favored by selection, at least in part because of that beneficial effect. So it's a general concept that applies to microbes, it applies to plants, of course, and it's not just an animal trait, for example, including requiring cognition or even communication. So it's a very general concept about helpful traits that are favored for that helpful effect. And so I'm going to explain this further in two sections of the talk. First, I'm going to talk about what has worked in the past, how cooperative crop plants have been used in the past, and some of the limitations of that. And then I'll talk about a new strategy that actually exploits potentially cooperative adaptations from nature. So first of all, I need to acknowledge that natural selection will offer favor to selfish and competitive. So if we, for example, look at this group of small non-competitive plants. Okay, I've put this gray triangle, if you can see that, to indicate, for example, that the shadow coming from these plants, or more generally just their zone of influence, how much resources they're using up. So in this case, these plants are not really having a harmful effect on their neighbors. And so that's why they're all, happy and enjoying high success. But imagine if a more competitive variant arises, and if that variant is often living with shorter, less competitive plants, then it's going to dominate, have higher reproductive success, and become common in the population. So that more competitive type can become common, even though there's a lot more harming, a lot more competitive effects going on in this new population. So even though there's more harming and group productivity declines, those selfish, more competitive traits are still favored. And so that's sometimes known as a tragedy of the commons, where individual selfishness leads to low efficiency and productivity. And that's what's thought to have happened in early agriculture, in early cereal agriculture. There's good evidence that the height of plants sharply increased in early agriculture. And that's probably because what happened at that time, farmers would take seed from their crop, randomly mix them to produce the next crop in the following year. And so what happened is, if there was a more competitive variant, they would tend to be living with shorter plants that it could outcompete. And that drives up the height and reduces the productivity of those crops. But in contrast, evolution is much different when genotypes can be grouped together so that similar types tend to live together. So again, imagine if we started with this relatively competitive population, if an even more competitive variant were to arise, and if it usually lives with similarly highly competitive plants, then they're all going to suffer from that competition. They're all going to be suffering from the harm that they cause to each other. And so that kind of strategy can't be favored in this case. But on the other hand, if a shorter, less competitive type were to arise, and it tends to live with other shorter, less competitive types, then they can all reap the benefits of that reduced competition, that relaxed competition. So that's really, in simple terms, how the evolution of cooperation works. So that can happen in nature, and I'll talk about that later. But of course, we can emulate this and do the same thing artificially in plant breeding. And in fact, one of the greatest successes in the history of breeding really adopts that simple rule that we need to keep shorter, less competitive plants growing together with like individuals. So during the Green Revolution in the 1960s and 70s, we introduced dwarf varieties of cereal crops, and plant breeders had the intuition to know that when they're testing different types of dwarfs, they need to keep them together growing with other dwarfs, and not allow them to be outcompeted by a taller variant. So it's that simple intuition that's so crucial for favoring more cooperative, productive crops. So because this strategy was so effective, and it led to huge global increases in cereal production, we know a lot about dwarfism, all the genes and signaling that's involved in creating a dwarfed crop plant. But we know a little bit less about why those plants are successful, and what's the relative role of cooperation versus some other benefits of using dwarf. And so, in particular, if you read the literature, it's often said that these dwarfed individuals are improved as individuals. One reason is because they don't fall over in bad weather and rain and wind. But another common assumption is that those plants can save the energy that they use to produce a big stem and instead reallocate that to producing grains. So the idea is that it can throw a lot of fertilizer on a dwarf crop because all that resource will go directly into the grains. But the alternative hypothesis is that dwarfed individuals are actually deficient at gathering resources. So they have this short or a small sort of shadow of resource use. They're not good at taking up resources, and so they have little harmful effect on their neighbors. That's what allows us to pack them into dense crops and reap the benefits of that. So the second hypothesis really focuses on that benefit of cooperation, reduced competition, whereas this hypothesis is more about direct benefits to individuals. So prediction that comes from these hypotheses then is that if we could grow plants in isolation, so in the absence of competition, by hypothesis one, you'd find that the shortest plants are able to reallocate energy to producing more grains. So their individual yield would be higher than taller individuals. But in contrast, under the second hypothesis, the smallest plants would be deficient in gathering resources. And so they're going to have less individual yield than taller plants would. And to take this prediction a step further, if we could detect this expected negative effect So where we see the benefit of dwarfism on group productivity and the cost of competition. Then under the first hypothesis, there'd be no trade-off. So what I mean is what's good for the individual would also be good for the group. But under the second hypothesis, there is this trade-off because here, what's bad for individuals, being short, being bad for individuals, is now actually good for the group. So I was able to test these ideas with a fairly new genomic resource called the NIAB Diverse Magic Wheat Lines. So NIAB is the National Institute for Agricultural Botany in England, in Cambridge. And they've designed this population. And I guess the important thing to know about what a magic population is that the M stands for multi-parent. So the key thing is that there's 16 parents to this population, 16 different wheat varieties, and they go through multiple steps of recombination and then multiple steps of selfing. And what you end up with are these homozygous inbred lines that are like a chimera of the 16 parents. So each line is a unique mixture of those 16 parents. And the parents of this population that I'm using are plotted here by their cultivar release date and a measure of the average height of those plants. So for my purposes, this was really useful because they come from pre-Green Revolution and post-Green Revolution. And I cover that wide variation in plant height. It's going to be all captured in this population and even maybe expanded in this population, shuffled into these unique inbred lines. And so I grew in total about 600 of these lines in two different conditions. The first one is the isolation condition. So plants were grown in the absence of any competition. And that was done in a controlled condition at the National Plant Phenomics Centre in Wales. And then I also grew them in field conditions near Cambridge. And what was unique about this experiment in the field is that we grew the lines at two different densities. The first one was a standard farming density at about 300 plants per meter squared. The second density was twice that, so 600 plants per meter squared, to ask if there is an appreciable benefit of growing dwarfs in especially dense populations. Okay, so here's the first part of the prediction. Remember, following plant height in isolation versus individual yield. And the question is, what direction will this be, positive or negative? And it turns out that it's quite fairly positive. Okay, so these shortest genotypes appear to be deficient at collecting resources. When there's no competition, they have all the resources available to them, but short genotypes just don't turn those resources into higher individual yield. And one benefit of using this population is that we know that that variation in plant height that I showed is highly heritable. And actually we can see clearly that it's explained by two key genes. So these peaks on this figure just show, indicate where in the wheat genome is a lot of that variation in height being explained. And it turns out it's these two well-known reduced height genes that were favored during the Green Revolution. So now you want to ask, can we see the positive effect of those dwarfing genes on the yield of of these plants grown in crop conditions. And this has actually always been a difficult thing to detect, because some of you might know, we do a field experiment. Some plots are just going to be in better condition than others. Some have more resources than others, even if you're trying to control that. So when you find a plot that has high resources, those plants will tend to be taller, and they'll also tend to have higher yield. So that kind of variation often obscures any negative relationship between height and productivity. So what I was able to do in this case is control for that variation in the field. So I'm going to ask, using a multiple regression, I'm basically asking, for all these genotypes that grew to a particular height in the field, what's the effect of having a shorter height in isolation? Does being a dwarf plant that grew to that height lead to higher productivity in the field? And so it turns out that we could detect that expected negative relationship. So overall, short genotypes or dwarfing genes are bad for the individual, but good for the productivity of groups. And moreover, we found that this benefit of dwarfing genes is particularly evident in really high density crops. So by growing crops at twice the density of standard and growing the very shortest genotypes possible, you get a fairly large boost, so let's say about at least half a ton per hectare in yield. So in general, this work supports this idea, a fairly old idea from Colin Donald, and that was that there should be an ideal cereal type, an ideal idiotype, he called it, for high density cereal crops. And that ideal type would be a very weak competitor. So it would be short, have small erect leaves, so very small window of resource use. And those plants might even be so weak that they perform poorly when grown in isolation. So we have strong support for that idea. But what about when it comes to making a recommendation for farmers? What this would lead to is a recommendation that we should use even more deficient plants at even higher densities. And so it really doesn't get past this problem of high intensity farming leading to lower diversity and potentially more inefficient use of nitrogen fertilizers. So I think it's helpful then to reevaluate other ways that we can make this shift from competitive crops to more cooperative, productive, and efficient crops. And beyond artificial selection, what I've said is that natural selection in special circumstances can also favor cooperative plants. So in the extreme where you would expect this is in cases where plants expand by modular or clonal growth, so that neighboring stems are genetically identical and they all form part of this large collective individual. So in this case, these neighboring stems should have been designed by millions of years of natural selection to be highly cooperative, highly efficient, all working together for the benefit of this collective individual. It's just like cells in our body all appear to be working for the same function of promoting the success of the entire body. You'd expect the same thing from a clonal or modular group of plants or group of stems that belong to the same individual. So this kind of cooperation could actually be vastly more powerful than anything that we can do by artificial selection. But because all these stems are connected physiologically, and natural selection has been working for so long to promote traits uh, that allow those stems to cooperate and act efficiently together as a group. So it's this kind of logic that led me to propose a new ideal plant type. So instead of being a small wheat competitors, actually what we need is to promote this kind of modular or clonal growth, get large individuals and plant them in low density conditions. So this is really a contrast to Donald's idea of a communal idiotype. So whereas that idiotype was designed for high density crops, this new colonial type is designed for low density. So obviously there's many stems in this case, whereas Donald suggested that only a single stem would be best. And whereas the idea was to design plants with short stature, small leaves, etc. In the case of the colonial idiotype, for now it was unclear what kind of traits would be associated with it because the design of this type is not up to us. So the design comes from natural selection. And so one of the questions that I was asking is, what would this colonial idiotype look like? So what kind of traits would be associated with that? And the other question, of course, is just how productive could this strategy be? So if you were to grow, we find an ideal plant type, a colonial plant type, and grow it in low density, what's the total production going to be like compared to a more conventional crop of small wheat plants grown at a conventional density? So in this case, this is what I've been working on most recently, and using another unique resource is called the Watkins wheat landrace collection. So these landraces have been collected in the early 1900s, so far before the Green Revolution, and this is when before really modern breeding had taken place. So you can see that this is a useful resource for me because this is an example of a single plant grown in the absence of competition. So these landrace plants are really big. They can grow up to 150 fertile tillers or fertile stems on a single individual plant. And so we wanted to ask, what's the best type to use in a low density crop, and what's the total production of these crops? What does it look like compared to a conventional crop? So again, what I'm using here is another kind of multi-parental population. So one of the parents of this population is a cultivar called Paragon, which is just a UK wheat type that was used after the Green Revolution. So it's a fairly modern UK wheat. And that's crossed with another parent, which is various lines from the Watkins landrace collection. So from those crosses, I used about 230 individual recombinant inbred lines. Again, just for the purpose of having a lot of variation. So a lot of variation in plant size, leaf size, etc. And the experiment was to grow these different types at 3 different densities. And so the standard density was about, again, 300 plants per meter squared, the typical farming density. Medium was using about 30% of that seed number. And then the low density was using less than 10% of the seeds that we'd used in the standard density. And so in the field, this is what the field looks like from above. I would have these triplets of unique Watkins genotypes with the 3 density treatments randomly assigned. And then you have this other sort of unique part of the experiment where I grew a subset of those genotypes in isolation. So the idea again was to grow them in an environment where they had experienced no competition from their neighbors. So this is what it looks like in the early season, and then the plants get very big later in the season. So the idea was to find the genotypes that do particularly well at low density in the field, and then ask, what is it about those genotypes? So what do they look like when they're grown in this condition where they're free from any competition? So eventually, for example, plant breeders could grow out their progeny in this kind of low competition environment and be able to pick out the types that would do well in a low density crop. So the first thing I want to show here is just focusing on the y-axis for now, the total grain yield of genotypes at medium density relative to the grain yield of that same genotype grown at the standard density, so in the adjacent plot. So how well do plants do in medium density versus standard? And so here's the data, and you can see that the average is about or close to 95%. Okay, so on average, even though we're using just 30% of the seeds to create those plots, they're producing above 90% of the total yield that a standard plot would produce. And so that's on average, but notice that there's also a lot of examples where this value is above 1. So the plots at medium density are actually out-yielding plots at standard density. Okay, so now I'm going to reveal that on the x-axis is that same kind of measure, but for the lowest density plots. So it's the grain yield of genotype at low density relative to that same genotype at standard density. And so in this case, the average is about 75%. So it's still quite surprising that when you use less than 10% of the seeds to create these plots, what you get out of that in terms of grain production is about 75% of standard. And again, there are also cases where that's much higher than 75%. So I really wanted to focus on these special cases where you have higher than average production in both medium and low densities. So I'm going to call these the all-star genotypes, so the low density all-stars that are really of interest to figure out what's so special about those that can allow them to produce so much grain at low density. So what I'm going to do then is I'll separate the genotypes into those all-star genotypes and all the others. And I'm going to look at the traits of those genotypes when we grow the plants in isolation. So I'm just going to ask, what's special about those genotypes and what could we measure about them when those genotypes are grown in isolation? So for example, measuring the flag leaf length, we found that those all-star genotypes had longer flag leaves, almost by 3 centimeters. So this is the average difference and the confidence interval around that average. So almost 3 centimeters longer leaves. And they're also taller, on average almost 10 centimeters taller than all the others. They did have more tillers, so more stems, but this wasn't actually a very big effect, and there's a lot of variation around it, so not a really surprising effect. But interestingly, they had higher tiller density, so the really productive genotypes at low density had a lot of tillers packed together into kind of small packages. So they weren't the big expanding genotypes, but the more compact ones. So what this allows us to do then is tells us that we can fill in more of this table, and it really emphasizes the difference between these two idiotypes. So it continues to be that the colonial idiotype is almost the opposite to this conventional communal ideal type. So rather than being short and having small leaves, well, the ideal type would be these tall plants with long, floppy leaves. And so now the final question then is, what's the overall productivity of a crop using this ideal colonial idiotype at a low density? So first of all, to compare with, I'm plotting this baseline productivity of the Peregrine cultivar, which is one of the parents of each of these genotypes. So clearly there's an increase in yield with increasing density in this Peregrine baseline. Now I want to just compare what these all-star genotypes look like in terms of total yield per plot. And so what you can see is that compared to the baseline standard at conventional farming density, there's a lot of cases where these genotypes are out yielding that baseline. So even at low density, remember, these are plots seeded with only 8% of the seeds of a standard density plot. One of those genotypes is already out yielding the baseline control at standard density. And then at medium density, so using just 30% of the seeds to create the plots, there's several interesting genotypes that are out yielding the control at standard density. So this is pretty exciting to follow up on, and that's what I'm doing in my work now, is just trying to figure out, are these genotypes consistently high yielding and low density, and what is it about them that makes them so special? And the other aspect of my future work is to take this new colonial idiotype in low density crops and ask, is it also productive and sustainable? So one question is this new crop design also productive in new environments? So is it productive in, say, Nova Scotia or Canada more generally? Another question, is it also equally productive with fewer inputs? Are those big cooperative plants also good at dealing with stresses like low nitrogen input? Another question is, are these large plants at low density ideal for more diverse crops? It seems like this new colonial idiotype would be ideal for designs where we're using intercropping to create a more diverse crop, and allowing those low density, or the wheat plants at low density, to thrive with another crop with it. And then finally, I'm asking, could this type be more resilient to climate change? So this summer, I just started a project where we're using drones to closely follow the elongation rate of these plants and asking, are the large plants in low density less sensitive to changes in temperature than smaller plants grown a typical high density crop. So that will tell us if this new idiotype is actually more resistant to a warming climate. Okay, so if you want to read more about the ideas in the talk, that this is a paper that explains this in more detail. A lot of the ideas that I've been talking about are known as kin selection theory. And William Hamilton is sort of the father of kin selection theory. So I developed this research program called Hamiltonian agriculture, which is just the application of this view of social adaptation applied to plant breeding and sustainable crops of the future. And so, like I've said today, I think this viewpoint is useful for understanding successes in the past, but probably the most benefit we can get from this is also recognizing that cooperative and productive plants have also been or should have been produced by natural selection, and we can actually exploit those plants more than we ever have in the past. And so the motto of the John Innes Center is unlocking nature's diversity. Actually, what I'm suggesting is more like unleashing nature's diversity, allowing plants to grow to their full potential, exploiting these cooperative, efficient, productive traits for more productive and sustainable crops of the future. So these are the people who support my work, and thanks everybody for coming.
So we've got 10 minutes. If we can have questions for Jay.
Sure, Professor Caldwell.
OJ, great talk, very challenging. I'm not sure where to start, but perhaps maybe we can look into some of the minutiae of it in terms of physiology and whatnot. But to come back to the whole idea of competition. I mean, and you at the end you touched on in terms of intercropping. I mean, a lot of the assumptions to what you're doing is that it's monocropping is part of the answer. Yeah. As opposed to a more complex biodiverse intercropping type of system, where, I mean, in terms of defining that competition, whether it's root or leaf or resource or whatever, are there ways of adapting your process to evaluate intercropping system?
Yeah, I think, yeah. So I'm talking mostly about the potential for reduced competition and cooperation among like genotypes, but the same idea is possible between species. So you can have complementarity or yes.
Because that evolves you along, right, in terms of how things interact and whether it's nitrogen fit. I mean, in terms of root structure, whether they compete or don't compete, what nutrients are taken up, those sorts of things.
Yeah.
To me, that would be a fascinating way of looking at the evolution.
Yeah, and so we could definitely screen different combinations, different genotypes from the two interacting species. And again, we could search for the most complementary or cooperative combinations and select for that.
Because that is the question of this cooperative or complementary.
Yeah, so I think cooperative would be would evolve over long term. I think that would be hard to replicate in an artificial.
But how do you see the plants, the wheat plants, cooperating? I mean, using the word cooperate. That's.
You mean within the
the system you're talking about? I mean, is it just less competition, or is it true cooperation?
Well, in the first case, where we're growing dense plants and we've artificially selected for dwarfism, for example, that's simply reduced competition, which is a form of cooperation, as I've defined it, but it's very simple, right? It doesn't depend on any connection between the plants, so that depend on any communication or anything. But growing those large modular clonal plants, you allow for a higher level of cooperation, because now you do have this physiological connection between the modules or the stems of the individual plant. And there, I think you'd see much more sophisticated cooperation, because selection has been favoring traits that would promote the entire collective fitness of those plants. So you'd see things like, for example, if one stem is infected with a fungus, you might have that stem become specialized in producing antifungal factors that can be shared with the rest of the plant. Where some stems are specialized photosynthesis in the shade, others are specialized in photosynthesis in direct sunlight. So there, I think you would see instigated cooperation that we don't fully appreciate.
So I'm not a plant scientist, I'm an animal scientist. So what are you, what Claude has brought up this question I know as well. So are you proposing that rather than have single plants grown in the future, we should have these clonal groups? That we would produce monocultures of these clones rather than.
Well, what I'm suggesting is rather than having a dense peer group of many individuals, so the idea is to promote less competition among plants. Instead, we could have these large modular clonal organisms where we have cooperation within the plant. So rather than promoting cooperation among plants, we have crops where there's these little pockets of cooperation within the plant. So we have these large modular plants growing at low density. So giving them space to grow into a crop. And it's that low density that would allow you to use intercropping, for example.
But then they wouldn't be separated. They would be then competing perhaps with those other crops you put.
Right, and that's the question of how do we make that work? So what's the best type to use in that intercropping situation? Like Paul was mentioning, can we screen different types, different species to even promote high complementarity among those species? But the key is that we need productivity in the primary crop. And there's more than one way to do that, more than one way. It's not just about these dense monocultures. We can get that kind of cooperation and efficiency in a new way.
Jay, I want to echo what Paul was saying. Thank you for your great statement. I have one comment about you mentioned for the short plant genotypes may likely allocate more resources to the seed development than to the high yield. So my previous work working on UP, and I found that under the stress condition, the genotype with the smaller seed size, they tend to do well under the stress condition. One of the explanation I think is under the stress condition, the small seed size genotype may more focus on the reproductive development. I have one question about you mentioned that there's a lot more noise for the grow plant under the few conditions you mentioned about some area they have more fertilizer, more water, more resources compared to other. That is why we have the randomization replication under the field condition. Do you think it will regression wasn't this to me? There's not very clear pattern for return for your progeny of your magic population. to me, the regression trend is not very clear. Do you think there's a chance that for the progeny of the magic population, the trait they continue to segregate? So the genotypes, they are not in the homozygous state. So that is why you mentioned there's a lot of noise when you grow the plant under the food condition.
No, I don't, it doesn't come down to heterozygosity. Those lines have been self enough that they definitely are homozygous.
So like seventh generation, like
More like 10.
okay, so
Um,
it's the homozygote.
Yeah.
Yeah, it should be stable.
Bring this up.
But I mean, can't think of the trigger you're thinking of that was all the variation, but I mean, I think you probably appreciate from any field trials that kind of variation will exist. We have, I mean, the way that we get around it is the benefit of using these lines is that you can have 600 unique genotypes, be able to detect those patterns, even though you have a lot of variation. So I think that's
Yeah, what a great resource you have. That's a huge resource for to do that experiment. Quite honestly, it's a difficult one to do. I think the fascinating thing to me was that 1900, the pre-Green Revolution plants, whether they in fact, I mean, all the Green Revolution plants post, I mean, there's a hierarchy on those tillers. In other words, the primary tillers outyield the secondary, the tertiary, and there is a real inhibition.
Yeah.
So from what you're showing, it seems like there's not that level of inhibition across the tillers.
Right.
Would that be fair?
Yeah, if they're given enough space to grow, then you don't see a lot of that latency in the tiller dial. So yeah, it's kind of
That's like under stress. If you come under stress, do they tell tertiary ones die first and that sort of thing?
yeah.
Yeah.
Yeah. And so there's a clear pattern where you see more of that tiller die up in higher density, less in the density. But when they're in the isolation environment, quite surprising that I think the highest tiller number that I have is 160.
And these are winter wheat. It's winter. There's
Well,
no spring wheat in this.
We grow them as if they're winter wheat, but a lot of them are spring cultivars collected from around the world.
But all those experiments were winter wheat varieties. Experiments, aren't they?
The Watkins, well, they are sown in the autumn, so we grow them in the winter wheat, but they would be classified as spring wheat. A lot of them. A fewer number of them are winter wheat. So these are, the collection comes from all across Europe. So yeah, some of them are winter wheat, the ones that live in the northern Ethiopia.
But you met here and got a baby. It's right. So
That's okay.
But there is a subset of the collection that is winter wheat.
Okay.
And so they.
Sorry, it doesn't mean sorry.
That's okay, literally. I am gonna cut you off so we can let others go in case they have a 1 o'clock, but you're welcome to follow up but thank you very much, Jay, for spending time with us today. We hope we see you again this month.
Research.